transactivation domain, one called the ATA, another called CTA. So what's that syndrome? Okay.
Just say okay. Sorry.
Okay.
I'm sorry.
Okay.
Yeah.
I'm trying to go through. They don't move it.
Oh, this one was working. I hope it's good enough so far. Okay. Or do you want me to just say click and I can advance?
If you can do that, maybe. Next slide. Okay. So how FLAG1 linked to autoimmune disease, and there's a rheumatology in the lab. He found working with Papas, he found that the overexpressed FLAG1, the messenger, is correlated with the lupus disease activity, and also in the human from the Diamond lab found that the increased expressed FLAG1 is associated with lupus nephritis. For most direct evidence from the overexpressed FLAG1, just 2 to 3-fold the protein will drive the normal streams to develop lupus disease. And they have autoantibody, they have renal deposition in the complex, and the mice will be died in the 6 to 8 months. But initially, this is the Dr. Allen Bursted's level. They are studying cancer. They thought they will be overexpressed FLAG1 will drive the mice develop some cancer, but instead they developed autoimmune disease. And when I came to MSc, and a couple of years later, working with Dr. Gary Gilson, and he gave me an opportunity to work in my own project. I pick up the FLAG1 I worked before. I say where I started, and Dr. Dimitri Spousis is here. He created this knockout mice FLAG1. If you completely knock the FLAG1, the mice will be died in the embryo stage. So what I do, I say, okay, if you overexpress FLAG1 and they drive mice, normal mice become autoimmune disease, how about I just reduce the FLAG1 expression? Or they will be impacted the lupus. So we've started with some autoimmune disease model. This one called AZB 2014 mice. These mice will develop lupus, but the later stage, like start at 23 or 24 weeks. So what we did, we backcrossed the B6 background with the AZM 2014 mice. We created the heterozyte and the wild type. And you can tell the heterozyte mice, the FLAG1 protein, top grade 1, will be reduced about half, but mouse will develop normal. So this is a piece of data take more than 2 years, because when you publish data, they always question you, did you have congenital? And we have make like a 12 backcross with the heterozygote. So what we found is amazing. You just reduce FLAG1 about 50%. The mice majority, like 93% mice, will be survived up to about a year. And in control, only 35% the wild type will survive. They make a huge impact. And if you check the lupus is kappa gamma immunoglobulin year, and they have huge activity of the B cell. But if you check the wild type compared to heterozyte, you see their total IgG and the GG1 to A. IgG3 is significantly, dramatically reduced, more than half in some stage, but the IgG2b is not affected. At some point, rather increased a little bit. So then, when we start the lupus, we starting the kidney disease, and you see the proteinuria in the kidney compared to the wild type. That's a blue bar. And the heterozygote will be almost dismissed. And then you check the pathology score, you see that dramatically decrease the pathology score. This pathology score may be not impressive because the wild type some mice died in the study. You cannot collect the kidney. You lose the probably most severe the kidney disease. And if you add that up, probably would much huge difference. And that's why they're showing about a half decrease. So when we have these mice, and people already question, and this may be just apply the one strain of mice. Then we're using another model for the mice called the MRL mice. We backcross another 2 years and created the heterozygous mice. But this is more impressive. And you reduce the FLI1 to half, and the 100% mice will be survive up to almost a year. In contrast, only about 25% mice, wild-type mice will survive. And this is probably mostly impressed profound the survival with any knockout the MRL mice we can find. So here also that we compare the serum IgG, you can see in the 8 weeks, this MRL mice develop lupus more quick than the AZB 2410 mice. You can see significantly reduce the total IgG. And in the 22, 24 weeks, and again, and we lost some mice, they died earlier. The MRL LPR mice start die, the female start die about 15, 16 weeks, and we lost that mice. And we probably should add whatever, at the age they die, and they will make the 22, 24 more significant difference. And we check the kidney pathology, you can see huge difference in the wild type and the FLI1 heterozygote. So then we start to counter what caused the kidney damage. One is mechanism is they have a lot of immune cells infiltrated to the kidney. We start to counter the wild type and the heterozygote. You can see they have significantly reduced T cell infiltration, neutrophil infiltration, and macrophage infiltration, and also B cell infiltration in the heterozygote compared to the wild type mice. And then this part of data we never published, but we thought, okay, FLI infect these immune cells. How about they maybe infect their development? So we create a conditional knock mice. We're using CD4 create and can knock the FLI1 beautifully. I have the Western blot I did show here. But if you knock out the FLI1, the T cell in the centers, the double single positive, double negative, double positive cell population are not affected. And also you check the spleen cells and CD4, CD8, the population almost the same. This is B6 background. So in a normal setting, if you just knock the FLI1 in the T cell, they not likely affect the population. But we all know if you're in the disease setting, and they will be affected dramatically, like the lupus. I think Dr. Xuezhong Yu at the microbiology immunology department, we collaborate. He has wonderful data to show how the FLI1 deficiency affect SDVs. And then we start think about, so why knock down the FLI1 or reduce the FLI1 be affected already? We start thinking about, maybe there are something drive the cytokine, because cytokine and chemokine are very important to regulate the immune cell infiltration. So the cytokine, basically chemokine, can divide it by 3 different. They decide depending on how many cysteine they link. And you have CC and CXC and all different this structure. So we focus on the 2 cytokine first, called the CCL2, also called MCP1. Why we focus this MCP1 and another called CCL5? Because this one show is very important in the lupus, and there's the urea lupus patient has high MCP1 and blood serum level MCP1 is higher, and also CCL5. So what we did is this is the work most done by a postdoc called Eiji from Japan, Suzuki, and Eiji did what he measured the MCP1 in the heterozygote and the wild type, and you can find that the heterozygote the MCP1 are reduced about half compared to wild type. And then his status kidney, they found that the glomeruli have huge MCP1 in the disease of the mice. And then he checked the kidney level of the MCP1 and the Rontis. Sometimes I try to use the same words, but maybe confuse you. Rontis also CCL5. And you can see the kidney's pressure in the heterozygote mice is reduced more than half compared to wild type. That's the message, that's the RA level. So then AG did, what he did, he using SRA FLAG1 can knock down the FLAG1 protein, the first two lines, you can see that. And the 3-4 line is control message RA. When you knock down that, the FLAG1 is in the endothelial cell. And the reason we choose endothelial cell, and at the beginning we found that the endothelial cell have very high pressure for FLAG1. So if you knock down the FLAG1, you stimulate the cell with the LPS, you can see the cells transfect with FLAG1 specific SRA has dramatically reduced about more than half the MCP1 production following the LPS stimulation. And then another postdoc, Mero, pick up the project with the CCL5. And he did the same thing. If you knock the FLAG1, you can see the cell production is reduced with CCL5. And then he did the CHIP assay. Everyone familiar with the CHIP assay. And because the FLAG1 is transcription factor, and they say, okay, is FLAG1 binding to the promoter of the MCP1 or the CCL5? And yes, indeed, he found in the CCL5, there's only one side that the FLAG1 can bind in there, but in the MCP1, there's 3 sides, and we call CHIP3, CHIP4, CHIP7 side. The FLAG1 can bind in there. So then what the Merrill also did, okay, how the FLAG1 drive the promoter and transactivation? And he cloned the promoter into a plasmid, then he just transfected the FLAG1. You can see in the CCL5, if you give as little as 0.025 microgram FLAG1, they can drive the activation significantly higher compared to the record only. Same thing with MCP1. So that's another transcription factor called the CSCL2, and he also show if you the bottom line you see the protein, you give a different amount of plasmid of FLAG1, you can see the protein start increase. At the same time, you drive the activation as you increase the FLAG1 protein. So both FLAG1, both the MCP1 CCL5 can possibly regulate by FLAG1. You have more FLAG1, you have more MCP1 CCL5, but the mechanism is look like a different. Remember, FLAG1 is the transcription factor. They have S domain, they can bind into the S domain. And the Dr. Maria Trojanowska, and she's in Boston University, but she works in the MSc. Her lab created the FLAG1 mutate, and they changed one amino acid from FLAG1 S binding site, 321. By changing this one amino acid, the FLAG1 will be lose ability to binding to the promoter. So when we need using the mutated FLAG1, then you can find in the CCL5, they are almost lost about 18% the ability to drive the transactivation. So in other words, FLI1 regulates CCR5 mostly by directly binding to the promoter. Then we did a little bit of detail assay, and just like using different radii for the promoter, and confirmed that data. But in the MCP1, I remember we have 3 binding sites: type 3, type 4, type 7. FLI1 can bind in there. But if you do the different the promoter assay, this a little bit maybe different or will be reduced to different degree. But if you look at this one, if you make the FLI1 mutate, they cannot bind into the DNA. They only lose about 20% ability to transactivate the FLI1 activity. And that tell you the FLI1 regulate MCP1 largely by the indirect, probably, and interact with another partner. And actually, we published the paper, and they are interact with innet kappa B to drive the MCP1 activation. So we also did a lot of other chemical, like GM-CSF, granulocyte macrophage colony stimulating factor, GCSF-L6, CSCR2. I have not published data, CSCR10. We found all they are regulated by the transcriptase FLI1. So then I have another postdoc and Zhang. And Zhang, what Zhang did show is FLI1 not only regulate the cytokine expression, and also the FLI1 impact the immune cell migration to the inflammatory site. What he did is, okay, first we created the GFP transgenic MRL mice, and like I say, all this take around 2 years to backcross. So basically, you have MRL mice, and they express the GFP protein. Every cell has this very broad protein. What he did, he collected these immune cells from the spleen, and when the MRL mice had the disease, peak disease, something around like I think it's about 20 weeks, and then he will inject this cell in either wild-type FLI1 wild-type MRL mice of FLI1 heterozygote MRLPR mice. You can see if you give the wild-type mice to the wild-type, and 18 hours later, their not cell will be in the kidney. And this is a little bit dramatically, but we quantify the number. If you give the FLI1 heterozygote mice the kidney, and they have few, and if you quantify the number, I want to pay a 2 number. First, we just like look at the first GFP positive cell. If you compare first column and the second column, what that means? That means, okay, you give a same wild-type cells, and give first the black bar is give the wild-type mice receive the wild-type cell, compared to heterozygote mice, FLI1 heterozygote mice receive the wild-type. You can see they are significantly decreased, about say 70%, and that tell you because the heterozygote mice, the kidney, they don't have less information. They can track, so they track the less immune cell. But you compare the first and the third column, what that tell you? That tell you they are both wild-type mice. But you give wild cell from the wild-type, but the third one you give cell from heterozygote mice. And the hosts are same, but they also still attract a significantly less cell. That tell you the FLI1 expression in the immune cell actually impact the migration. And so we verified this one in vitro chemotax assay, and indeed the heterozygote cells have less migrate compared to wild type mice. And so if you look at this one, they have a lot of the chemokine have chemokine receptor. And if you affect the migration, likely you affect the chemokine receptor. This is one of my colleague, Tam Nolling, did this work, and he found a flower regulator, one of the chemokine receptor called CSCR3. And from T cell, you can see heterozygote mice have significantly reduced CSCR3, and they also, this is part of the cell where we have less migrated to the kidney. I would just like us to go to next the research work, and this work was just published this year, was done by Xuan and Wang, and she was a graduate student. Well, we start, okay, you genetically reduce FLAG1, you can reduce the disease. If FLAG1 can target that, drug can have some therapeutic development if that's the case. And indeed, Dr. Ben David, like I said, the first he and Dr. Dennis was it probably at the same time. And what I heard is Dennis said that he first identified it a little bit a couple of months earlier. Dr. Ben David identified the flower anyway. And what he continued work with flower with cancer. And he identified a couple of drugs that can inhibit the flower. And this is the data from his publication in 2012. You can see there's a drug called topo inhibitor called camptothecin, and can, if you give very low dose, in this case like a 0.05 micromolar, can significantly reduce the amount of flower. So the camptothecin was used in China to treat some cancer, but in the United States, and they wait to the phase 2 clinical trial, they did become FDA approved drug. But the same class later they made a modification called the topotecan, and was used to treat I think is ovarian cancer and lung cancer in the United States. And there's another drug called the erythrocan, and they also was approved by the FDA. So what we did, okay, we did using a model called the AZB WF1 mice, and we started to treat the mice about 25 weeks. And at that time, about half mice have a limited proteinuria. And we have 7 groups of mice, and either have a high dose CPT, low dose CPT, TPT, topotecan. We have 3 groups: high, middle, low, and we have negative control, just give a PBS, and then we have positive control, cyclophosphamide. That drug was standard to use in treat the lupus nephritis that have some toxic. And the dose we decided is we based it on the treatment for the cancer. We're using about 1/5. The high dose is equal 1/5 of the dose for the cancer. And then you can find that all these treatments except the low dose of TPT, and they are 100% keep the mice alive. And then this is a lupus nephritis model. We measure the proteinuria, and you can show the first control mice, except one, all the 9 mice have very high proteinuria, and the CPT low basically, and very few have very low, and then we reach different, but TPT high almost very clear, and CPT high also very clear. And the CYC, the cyclophosphamide, have one mice develop proteinuria, relatively high, but all the TBT low also too low. Most mice develop severe proteinuria, and the topotecan middle range also, except the one have a little bit, but most are very clean. That's the 40 weeks before we sacrifice the mice, and we can quantify the number. You can see, the which one. The control is red one, and the square black is the low TPT. These two groups have a higher proteinuria. We can just focus on left figure, and also the cyclophosphamide have a little bit, other than proteinuria pretty lower in other treatment group. And when you treat the lupus nephritis, another thing is to measure the kidney function. In the left bar, we measure the called the kidney index. Basically, if you higher index means there's inflammation going on. You can see the compared to the first bar is control. All these CPT, high dose, low dose treatment, and TPT, middle dose, high dose, and also house control CYC, they have significantly reduced the kidney index. Another measure is measure the blood serum, the BUN, blood urine nitrogen. And you can see the same, except the low dose TPT, all other treatment groups significantly reduce the BUN. And also creatinine, I will measure that. Same result. And another thing is the lupus is a spontaneous autoimmunity. You have enlarged the spleen. You can see, the spleen size. We measure the spleen size, except the low dose of the TPT, and all the spleen size significantly reduced in all other treatment groups. And we counted the total spleen cells. That's in the top and red figure, and they are significantly reduced the total number of spleen cells. And we want to see the mechanism. You give the drug, and what's the mechanism? So we measure the Western blot, and we add up, quantify. You can see the same thing. The trade with the CPT or the high dose, a low dose TPT, and reduce the flavin expression compared to the control, but not the CYC. CYC is the same. So then we measure the double-stranded antibody, and you can see all the treatment group significantly reduced the double-stranded. And remember the low dose of TPT, and they are not they are not impact the saliva, but they are significantly reduce the double-stranded antibody. And then we measure the serum, total serum IgG, IgM. You can see most of the treatment group significantly reduced the total serum IgG or IgM. And they will measure the different subclass. I think this IgG1, IgG2, basically a same trait. And there are some class. First is that's the IgG1, IgG3. I couldn't see clear from here. So another way to measure the lupus nephritis is to see how immunocomplex. The basic the lupus is you have immunocomplex, and they deposit in the kidney, and they activate the complement that attract a lot of inflammatory cells and cause damage. And then we measure the total IgG deposition, and the C3 deposition will quantify the deposit. You can clearly tell, except the topotecan low dose, all these treatment groups significantly reduce the IgG deposition, immunocomplex deposition, and the C3 deposition. Okay, I still have time. Maybe speak too fast. Then we measure the kidney pathology, and this is done by Dr. Heck, and we do the double blind. You can see all the treatment group except the TPT lower dose, they significantly reduce the kidney pathology. So we show this because it's a chemo drug, and they always want to ask you, and maybe you know this is the really cyclophosphamide also chemo drug. They cause a lot of the side effect, and we measure the side effect. You can see the liver toxicity is no change, the ALT and no change. But we measure also the CBC count, and is a very interesting found. The CPT treatment actually not affect the red blood cell and lymph node. Actually, this group of mice treated with CPT has significantly higher than the CPT. Then we measured also the red blood cell. The one thing I'm still puzzled, I don't understand. If you look at the the tall bar is the CPT, high dose CPT, they make a significantly higher the platelet. The really I say I'm puzzled is because we show the CPT reduce the FLI1. If you reduce the FLI1, suppose the platelet will be reduced. But in this case, and the platelet number and is significantly increased. We have not figured out the mechanism.
What about the function of the platelets? Do the mice have strokes due to this? Because that's a really high number.
Right. We did not measure the function of the platelet, but we did not see any sign of the sickness of the mice.
Okay.
Yeah, and that's a good point. Yeah.
Is this a product of megakaryopoiesis or platelet release?
Oh, we don't know the mechanism. Yeah, that's a good point. We know the knocked-out FLI1 mice will have significantly lower number, most close to zero. That's the one that really mice died in the embryo because they don't have enough platelets. Yeah, but this is stuff, maybe they are overcompensated.
So there is another question from Wei. So great talk. Thank you first, right? May I ask what is the mechanism of FLI1 mediated inflammatory cell infiltration to the kidney, but not the thymus?
Not the thymus.
Yeah, you know, it goes to the kidney and not to the thymus. Why is that?
Not to go to the thymus. I think the kidney, what we show is, if I understand quite clearly, is because the kidney has inflammation. And when you have inflammation, you have a lot of the cytokine produced there. Then they attract this, the immune cell, infiltrate that. The thymus is no inflammation in this case, I think. Did that answer your question?
So where is it?
Oh. Yes. So do you mean it's not FLIP1 directly? It's indirect through inflammation.
Is both way, like we said. Oh, there I didn't touch anything there.
Remind me later if I can move, but yeah.
So no, this is both like if you reduce the FLI1 expression on the immune cells, there will be a factor there's ability to migrate to the inflammation side. At the same time, if you have a high cytokine production and in the inflammatory side, you will be attract these cells. That's answer your question.
So, but in the thymus, there's also lymphocytes. Do they express FLIP1?
Yes, the T cell highly expressed, but in the thymus, in the normal mice, and they not affect them, but they don't have the disease, they don't have inflammation. And in the diseased mice, we did not measure that they go to thymus or not.
I have a simple question and a more complex. So the simple question is, when you say CPT treatment or TPT treatment knocks down FLI1, what are you measuring FLI1 expression in?
We measure the spleen cells, the Western blot, yeah.
Oh, in the splenocytes.
Right.
Okay. So the more complex question is, the origin of reduced fly expression versus full strength fly expression, is the original phenotype one of hematopoiesis, or is it the phenotype of the immune cells on an even playing field, or a combination?
I think your question is FLI1 affect the hematopoietic cell development, and if that's what you want, we don't know that. And because they are chemo drug, they may affect some, but because we see the CPT treatment mice is phenotype is very similar to the FLI1 reduced heterozygous mice. We
Right.
think maybe affect is through the suppressed FLI1 the total protein.
Right,
We
and
get rid of this now.
it's, so focusing on FLI1 as a transcription factor and looking at downstream targets is one thing, but in all of us in this room and listening, what is it upstream of FLI1 that will change FLI1 expression?
That's a very good question. I will get some, not all, but let's say the network that I cannot go next now. Can go next.
Yeah, be the first one.
Yeah. Okay. Here, so let's see the glomerate. The lupus nephritis is a glomerate nephritis, and basically there are three cells having the glomerate, and you have podocyte, you have endothelial cell, you have mesangial cells. We don't know the podocyte, and we know the mesangial endothelial cells, and is present highly expressed FLI1, especially mesangial is very important for the inflammation and also disease development. Next, please. This works.
Can do
Okay.
that now. Yeah.
And then we use the human glomerate endothelial cells, and we treat with the CPT. You can see the A, and we're using different dose. You can see the FLI1 protein decreased. And then we stimulate the cell with interferon alpha. And you can see the B, if you measure the MCP1, if you treat with interferon alpha, you treat with CPT, they are produced significantly less than compare the DMSO visual control treatment. And the same thing, the CSCR10 also, if you treat with CPT because it reduces FLI1, I will show you that because it reduces FLI1, and they are produces significantly less CSCR10, and they also call the IP10. And then they will measure another human primary renal mesangial cells, and we treat with CPT or TPT. You can see, and they significantly produce the MCP1, and the TPT and they reduce less than the CPT, but also the difference was significant. Then what do we measure? Okay, I think Dimitri is also this part of question maybe answer it. Is the FLI1 really affect the CPT really affect the FLI1, reduce the inflammatory cytokine, and because the CPT also affect the chemo drug also affect a lot of other side effects. And what we did the experiment is this experiment. Okay, we have mesangial cells, human mesangial cells. We treat with the interferon gamma only, then we treat with CPT, and then treat with interferon gamma. Then we have another one, treat with CPT, reduces FLI1. We transfer back to the FLI1 expressed plasmid. We hope it can bring the FLI1 protein back to the previous level. And from the first, you can see, okay, if you have FLI1 black, you treat with the CPT, the third line, you will bring the FLI1 protein down. But if you transfect with vector only, you didn't change the FLI1 protein. But if you transfect with flavan plasmid express flavan protein, you will restore the level almost the same like you before you treat with CPT. CPT treated cell can last a couple of days. And then we stimulate them with interferon gamma and we measure the MCP1 and the CCL5 and 24 hours later. And you can see from the B and C, if you bring the flavan back to the normal, and the production level of CCL5 and MCP1 will be restored. And what that tell you the prove what we thought is CPT reduce the inflammatory cytokine mainly by the reduced flavan. So a lot of the data I talk about from here is non-published, and so probably can bring more interest to the everyone. So first, microglia. We know microglia is like macrophage cells in the brain. They play a huge role in the neural information. What we did is, okay, if we have human microglia cells, if we knock on flavan, we can see, we stimulate the cell with interferon alpha in this case. We can see significantly decrease the production of the MCP1, CSL10, L8 in the microglia cells. So then we tried another called the monocyte, and this is a THP1 monocyte, human monocyte. If we knock the flavan, also we stimulate with the interferon alpha, we see significantly reduced MCP1, CSL10, and Rantis CCL5, L6 in this case. So that's why my title say the impact of the autoimmune diseases, not only lupus. I think some of flavan involved like multiple sclerosis and potential to develop something by reduced flavan will be affect the multiple sclerosis. So I want to bring this piece of data, maybe Dimitri, released with a question, not directly, maybe kind of you will know what I'm talking about. So this is the endothelial cell. We stimulate with the LPS, so we collect the cell, and the line from A, the line 2 is like 1 hour later, 3, I think it's 2 hours, and then 6 hours, 4, and 24 hours later. And if you see the total of the flavan protein, they are not changed too much. But then you measure the MCP1, you can see the production is up. So what this tell you, I so far I keep it.
Can I interrupt you just for
Sure.
before Beshu is asking if you mean that the FLY1 controls on interferon responsive genes?
Is control is response? Yes. Interferon, yes.
Yeah.
Yes. Interferon, we have the data. Interferon alpha, interferon gamma, depending on which cytokine response. Yes, they are largely affected interferon gamma or interferon alpha and genes. We have not tried others. We tried both interferon gamma and interferon alpha. Yes. So far, I tell you, if you reduce the total flavan protein, and they are measured, it will produce less cytokine. But actually, from this piece of data, I also tell you, a lot of this activation, actually, like Dimitri's question, I'm not sure related or not. I think actually flavan's activation largely in the cell is through the probably post-translation modification, because like you have 10,000 RNA, you add 20,000, reduce 2,000, and there will be less fighting. But at the same time, you have 10,000 army, they don't have weapon, they also cannot fight it. And the same thing here, the flavan protein not change too much, but they have some modification can drive the activation. And the same thing here, this data. This is the human renal endothelial cell. And let's see the first two. You have this, the control SRA. You can see you have control SRA, and 0 hour and 24 hours after stimulation with interferon, you don't see the protein, only slightly maybe up, but not too much. Fly1 control, Fly1 SRI, you also see they are down compared to the control SRI. But at the 0 and 24 hours, they don't change too much. But if you compare the 0 hours, the CRCR10 and 0 hours MCP1 to the 24 hours, they are significantly changed. And this data also tell you the Fly1 activated mostly through the post-translation modification. So that's the, okay, I think I will put a summary. That's what I want to do next. Everyone know the story in kappa B. If the kappa B program more broad, every cell have in kappa B, but Fly1 is limited to the immune cells and endothelial cells. Also some fibrous, maybe, but not every cell have high expression of Fly1. But they also kind of have the ability to control the inflammation. And basically, I think Fly1 have 3 mechanisms to impact the inflammation or autoimmune disease. First, if you have stimulation with LPS interferon gamma, and the Fly1 will be acted in the local inflammatory side. In this case, for example, in the kidney, and they will be active the mononuclears in the cell cells, in the brain, they will activate the microglia cells, macrophage. They make these cells produce more cytokine. They can attract the inflammatory cells. Second mechanism, I think that they will be impact the immune cell migration, because you stimulate the Fly1 and they are Fly1 activate, they can make the cell more migrate through the Fly1 act, the example I show you, increase the chemokine receptor, they can make them fast to migrate to the inflammatory side. A third one, the mechanism is Fly1 impact the autoimmunity by impact the immune cell activity. We show if you induce the Fly1 in the lupus mouse, and the mice will be reduced to the total autoantibody, total IgG. And there are some, the mechanism we are not fully understand. And because Fly1, very few people starting, and I think that this is a drug, this can target by drug. The one mechanism we show you can directly hit in the Fly1 expression, like we show using the Fly1 inhibitor, but another mechanism to target the Fly1, we don't know how they activate. If we figure out which kinase and involved, we can target the kinase, block their activation, and then maybe can somehow have therapeutic effect on the disease. Okay, and the final slide, I want to acknowledge a lot of the ground support from the VAH and Lupus Research Foundation, and a lot of collaborators and the work done by the people working in my lab. And thank you very much. I would like to take a question. And,
Hi, John. Center.
Good talk.
May I ask a question?
sure.
Go ahead. You need to speak up.
Yeah, so I have a quick question. That is so in lupus patients and lupus prone mice, do you see higher level of the Fly1 expression in immune cells?
I'm sorry, what's the question?
The question is, do lupus patients and lupus prone mice show higher Fly1 expression?
Oh yeah, initially, if you look at our first paper, we compared the biopsy mice. We found there have the Fly1 is a little bit higher, significantly higher. We wrote down the paper, but I can tell you, the very honest I don't think that the purity level really matter. Probably about the same. But the most matters, I think, is because you have this inflammatory signal, like in the lupus, you have interferon, type 1 interferon, interferon alpha. I think that's drive the activation, probably not at the purity level. But if you just look at the first paper we published, we claim that, but I don't think that's really the case. Yeah.
So that leads to the question, like, can inflammation trigger nuclear translocation of FLI1, and it increases the nuclear translocation, or the FLI1 is always in the nucleus?
So here is the voice like a.
No. The question is if inflammation, for example, when you add interferon alpha or interferon beta to cells to activate, does that trigger nuclear translocation of FLI1? Or the FLI1 is always in the nucleus, it is active only during the immune activation?
Right. As far as I know, the FLI1 is always in the nuclear. They don't have the beautiful story like NF-κB. But you see the one paper Dr. Ben David published, he said some FLI1 located in the cytoplasm. I think that's a little bit of corporate background thing. But as far as I know, the FLI1 always located in the nucleus. That's a little bit harder to study. If you do the NF-κB, you see a beautiful story from cytoplasm to nucleus. I think that's the case. You have the single interferon alpha and all other signals. You bring the activated FLI1 just like a phosphorylation, acetylation. We have papers show acetylation actually affect the FLI1, the activation significantly. Yeah.
Yeah, so that's the
Thank you.
question of what post-translational modification is. So if it's not upregulated expression, what post-translational modification? And phosphorylation could change cytoplasm to nuclear and now have transcriptional activity. But you mentioned acetylation. So what would acetylation be doing?
Yeah, that's the way. That's my task. I want to figure out that. But there's a piece of data, and if you see our publication, we just artificially to show, because Maria Trojanowska's paper show some acetylation affect their binding, and we using the protein from her, we can see they affecting the transactivation to affect the promoter. If you change one acetylation or phosphorylation, yeah, we have that data. But in the real world, and if you simulate the interferon gamma, which gather, we don't know that.
Okay,
And that's what I want to figure out.
Okay, and as far as so chronic inflammatory diseases, obesity, and you talk about LPS, so you're talking leaky gut and maybe leaky endothelial cells. So if you look in animal models for that, do you see upregulated FLI1?
I have not seen that. I will be imagined probably the protein will not change, but the activation FLI1 will be changed to drive the local whatever leak to produce the cell to produce more inflammatory cytokine that attract more inflammation.
And do you think lupus patients are a combination of post-translationally activated FLI1 and overexpressed FLI1, or one or the other?
I think mostly from what my understanding, I show you paper, this paper show messenger RNA is parallel with the activation. I think most probably through the activation FLI1, the total protein level is measured by the lupus patient and the normal, also the mice, probably the level did not match too much.
Yeah, and then with the last question with the therapeutics, CPT is a topoisomerase inhibitor. So it sounds like it may be acting indirectly to regulate FLY1. Are there any more direct or direct down regulators?
Yeah, the mechanism, we don't know how, why CPT reduce the FLY1. The mechanism we don't know, but we know the CPT in the mice treatment maybe through inhibit the FLY1. The other directly, indirectly stop because they are chemical. The maybe, but we don't think that's the major cause to reduce this disease, because we show, you know, if you reduce using CPT back to the FLY1, they will restore the inflammatory response. Yes, but also, well, I want to emphasize the dose we use compared to cancer treatment is like a 1/4 or 1/5. The cyclophosphamide treat the lupus nephritis using the same dose treating lupus nephritis and the cancer and improve. But if we're using this drug like topotecan, FDA approved, you can use in 1/4 dose to treat the disease. I think we will have, you know, at least give another option and potential have less side effect. And if you figure out the other stuff can block the kinase or something, that's another pathway we can.
And if you
block
try
it.
combinations
Right.
to have even further reduced
Right.
dosage of each.
Yeah. So Dimitri, you already figure out what I'm writing the next round. That's what I'm writing. Yeah, I'm trying to figure out that to target this, not just directly reduce FLY1, target the activation for next potential.
Right, because you want to give this to the patient every day or.
Right, they are
your
in the clinical.
maintenance therapy.
Right.
Hey, John, so great talk. This is Zhang. I follow up the question. You mentioned that the post-translational regulation of FLY1. Do you know what way to be able to regulate FLY1 activity through phosphorylation or acetylation? What is the knowledge, existing knowledge here?
Yeah. We have done the promoter level. We just play around. We can see sometimes depending on which cytokine, if you phosphorylate the FLY1 and they are suppress FLY1 activation. And actually, acetylate the FLY1, you are drive like more than double their activation. The same amount of the protein FLY1, we play the promoter. But in the real world, we don't know. I talked with Dimitri, you know, I want to do the FLY1 protein mapping to stimulate the FLY1 with the interferon alpha to compare before and after, found which amino acid was phosphorylated and acetylate. Then we can figure out which kinase involved, which other enzyme involved. I think that's what we want to do. Yeah.
So phosphorylation is one of the way to regulate.
Phosphorylation is definitely involved. Yeah, I have a piece of paper in the T cell. I found mainly the phosphorylation is happening in the serum. Dr. Maria Trojanowska found in the fibrosis, FLY1 mainly phosphorylated with tyrosine. Yeah.
I have a second question regarding, I mean, you have a lot of autoimmune disease model and many use heterozygous T cells. Now, you know, you have the conditional lockout. Have you tried to compare heterozygous and homozygous FLY1 lockout T cells and in your disease model?
No, we have not tried that.
Okay.
Yeah.
So as you know, as a through collaboration, we see the heterozygous and homozygous deficient seems different impact the T cell pathology, pathogenesis, and I think it's interesting to looking at you have already so many well-established model, and that will be understanding more biology of how FLY1 regulate T cell response.
Yeah, that's very excellent question. I think like probably from like why you don't want to over drink too much damage, but also drink a little bit, have some degree will be happy. Maybe that's not the example, not a property, but you definitely, yeah, we saw your data is, yeah, it's huge difference with heterozygous and wild type, and also complete knockout. I think that's the case. You don't want to there. You want to suppress them to some level, but you don't want to completely get rid of flan because they have lots of other good side. Yeah, I think the moderate is worse, and they will, and it's a little bit complex because transcription factor they affect so many different downstream genes, like Dimitri said. Yeah, but is you can be targeted and our data show yes. Thank you, Chip. Very good question.
Yeah, thank you. Well, thank you for your insight. Okay, I think we are past the time, and really thank you for your great talk here.
Thank you. Thank you, everyone.
Thank you so much.
All right, thank you all for coming.
Thanks. I came running down here.
And
when
thank
I-
you for agreeing to the
I got
time
you.
change.
That's good to see.
All right, see you all
Very
later.
good stuff. Very, very excellent work. You've taken it so far.
Yeah.
Yeah, and the idea that you could actually help